So I'm working in a HPC center in the University of Milan. And today I'm going to actually not speak about mean field models, but I will actually start from the micro scale. And as my title is stating here, I will work at the cellular level. And what I want to do, it's basically what it's summarized here. So we have the anatomical connectivity. We have seen several applications of data analysis of multiple cell recordings, and you can infer about how the information is flowing through the networks with functional connectivity tools. But there is also another concept, the effective connectivity, which attempts to find causal relationships between neurons. And that's the way to go if you want to better infer from activities the anatomical connectivity, right? Because with functional connectivity, you can frequently go wrong because you have indirect effects. You have many neurons that interact in the network, and just relying on statistical relationship, it's not. Often you go wrong. And so this is basically the focus of my talk. So I will speak about spikes and PSP. So these will be ePSP and iPSP. The approach is maybe still a little bit abstract with respect to what are the possible recordings nowadays. So, I will just here mention what we can obtain with multi-electrode recordings and with calcium recordings. So there are, of course, many things we can do, and we had a lot of talks these days. But still, there are some issues in interpreting some part of the signals. For example, in extracellular recordings, you have issues with spike sorting, and in very high-density recordings nowadays, you have the problem that a single cell can be recorded for multiple electrodes. In calcium recordings, yes, you have, of course, the slow decay of the calcium signal, which sometimes make it difficult to interpret when you have several events that are summing. And I will just have a small detour here because I'm involved, I'm working with Marcello Massimini also on PCI, and since PCI have done this work, I would like to mention that because it goes in the direction to explore what is the minimal requirement of a network to display complexity. So what I did here in a collaboration with a former colleague from the Italian Institute of Technology, we used cell cultures, and we exploited the idea that, well, these networks, despite being very simple compared to real tissues, they have these small clusters, so they have some prerequisite of complexity, they have modularity in the network, and of course these modules are communicating, so they have long fibers connecting the modules. So apparently, based also on the idea of the theory of Tononi, they have the right requirements. And what we did here in this study is to stimulate, so to take the control condition. In the control condition, cell cultures typically display a firing regime that resembles the sleep-like behavior. And what you can do is to add drugs and see how the network is going to react. And you bring it farther away from the sleep-like state. You try to obtain something that resembles to the wake state. It's not really easy stuff. But still, I mean, you obtain something which is much more closer to the wake state. What we found in this study is that actually PCI is not really changing that much if we apply carbachol, so it's not at all comparable to experiments done in slices by Mavi in several studies. So you have just this small increase, which actually is even not significant. And this is to say that, yes, maybe if we want to start from the ground, we need some more properties that maybe can be found in a three-dimensional cell culture or in systems that have a bit more constraints. So that was just a small detour, just to, to say, well, there is some other activities in HBP at the micro scale that we have done. And coming back now to the topic of my talk, so what I would like is to link to find a way to use effective connectivity to infer the connectivity of networks. And what we basically need, as I was saying before, it's some info, some hints about the ePSP. And we know that these events in neurons can be very small, on the scale of 0.5 millivolt. And there are nice techniques also. It's really a niche, so there are not many people using those type of signals, but they're really nice, these genetic encoded voltage indicators. Here in this picture, this is taken from the paper of Gon in 2015. And here they show they compare electrophysiological recordings with fluorescent recording. And you see that you have really the same spikes, and the precision and the time in the timing of the action potential is on the order of 1 millisecond, so it's very good. And you can even resolve these SACs in the subthreshold regime, which are on the order of 5 millivolt, which is probably still not enough for resolving spontaneous event and EPSP, but let's suppose we can use this kind of technique or similar technique and isolate and, detect EPSP in our recordings. I should say that in order to detect the events, you need, of course, also good algorithms to find the events in the recording before starting the model and the analysis. And we have been working also on these aspects with filters that are called Perona-Malik, and we adapted one of these filters and we improved a little bit by increasing the sensitivity of this technique by 5%. So the proposed methodology, as you can suppose, it's based on information that comes from spikes and EPSP. I will distinguish the external from the internal process. External processes are just spikes that goes out from the neuron eye. Internal processes are all the events, the EPSP, that are received by the neuron eye. And I will consider in what's called an event-based model. So we will slice time in delta of 1 millisecond, and we will define just these binary variables. So it will be 1 if there is an event, so if there is a spike, or 1 if there, in this side, if there is an EPSP. And what do? I will not enter into the detail. It's not really tough math, but the essential ingredient here, it's, as you can imagine, is if there is an EPSP at time step n+1, it basically means that something is coming from the network, from other neurons, J, and we can make this binding the probability through a logistic equation that takes into account the potential contributor of the other neurons of the network. And well, we can plug it in a log likelihood function. And in addition, which is an important ingredient in the methodology, is to add a lasso penalization term, which basically it's weighting the complexity of how many weights you're putting in the network. And this is a good aspect because a nice approach, because the lasso penalization tends to promote sparseness in the networks. And since the biological networks are by themselves sparse, so we have many non-existing connections, so that we have never n-square connections in the network. This is, somehow the right way to penalize this log likelihood function. And what we do here is then to minimize this log likelihood function, and we take into account all the weights, all the beta from the process that are different from 0. As I was saying before, many will be set automatically to 0 because of this lasso penalization. And of course, the approach I have presented before is working for EPSP or no event, but it can be extended quite easily also in order to take into account a different class, which is the IPSP. And what you do then, of course, since you can simulate the activity and compare the real connectome, extract the activity, and you compare the activity, you try to infer the network from the activities, is just a comparison between the inferred connectome and the structural connectome. And this is something, the principle of this methodology has been already outlined before some years ago. And what we do, you compare the real connectome to the inferred one, so you build what is called a confusion matrix, and it basically has quantities like the true positive, which just tells you the inferred connection is existing, the false positive is just inferred, but it was not existing in the real connectome. And putting together, I mean, these numbers, you can define quantities like the sensitivity, the specificity, and build what is called the receiver operative curve. And basically, if you look at this curve, what is going on is that sensitivity is going very high at the beginning. So you have different points because you have different lambdas. So I'm testing different penalization of the weights. And it goes up to 1, meaning that the sensitivity is really high. So you have almost no false negative. And also the specificity is close to 1 because you have here 0. And that means that you have also almost no false positive. That is to say that, well, every time if you are familiar with the receiver operative curve, you know that this is really the best performance a classifier can obtain. And indeed, the area under the curve is close to 1, which is the maximum you can get from a classifier. What we did then is to introduce, as I just was saying before, also inhibitory neurons. I should say that we added much more noise in order to test the robustness of the methodology. This is to say that the methodology is really robust to the noise. So since you rely on just very few real events, and I will comment also on that on the end. And there are other metrics, so we repeat the same simulation as we did in the former work here with 20 neurons, and including also inhibitory neurons. And again, it works pretty well. So you have this ROC curve, which is perfect. And there are other parameters like this Matthew correlation coefficient that is used in the field when you want to infer networks and compare them to the structural networks. And it basically goes to 1, which is the maximum the metric can reach. And then changing lambda. So lambda is weighting how much you are weighting the inferred connections. So if you don't weight it, you have basically all the connections in the connectome. While if you start to weight it more and more, you see here many connections are disappearing. And this is because all this beta are becoming 0. And you are losing a lot of connections. So that's the way it typically works. Again, here, performance works good. Then you might wonder, is the methodology applicable to many cells? And indeed, we found that even with 100 neurons, the approach is working perfectly, as you can see here. And I should stress one point, which is really important also for experimentalists. So what is the time scale? So the time scale is 5 seconds. You don't need much more than this. Here the firing rate is maybe a little bit higher than in experiments, but you know, it's a matter of compromise. So lower frequency means you need probably a longer recording, but this is in principle saying that things can work even with a very short time window. And what happens if you scale it even more to 100 neurons? I should say that I did it only once because the library I have used is the really standard one. So you have this Shikit library, which is the entry point if you want to do machine learning using Python. It's really a nice library, but unfortunately it's not really efficient for this kind of problem. Of course, I discovered that later at some point when I already implemented everything. Here, for 500 neurons, you have 25,000 parameters, which is quite a lot. And in order to calculate this curve, it took me hours and days, so it's really not the way to go. Thankfully, this type of problems are really standard, let's say, so you have libraries that can scale up to 100 million parameters. So there's no problem technically if one wants to apply this technique. And another problem, which I think it's really, really relevant for experimentalists, is that when you have a network, you are never sampling the entire network. So you are sampling some neurons, and what happens if you take into account that you are going to sample just some part of the network? Doesn't the methodology work? Yes, it still works. So you can pick up randomly networks in this type of network, and still this MCC peak is close to 1. Of course, you have the fraction of inhibitory cells that changes because you are randomly picking up the cells. And I think this is quite relevant, of course, for experimentalists. And have I had just slides with random networks up to now. Now I introduce also topology, not because it's nice, of course it's more realistic, but also it's challenging, of course, because now you have larger variance in the connections. You will have neurons with a lot of connections, and others that will have very few ones. And still, the methodology is robust and it still resists. And also the test with sampling 20 neurons out of 100, so it's still working. This is an extreme case where you have just 2 clusters, few connections between the clusters, and again, the methodology works pretty well. Now, there is a concrete problem, and I'm almost closing my talk, so I have 2 slides. So we can go to the coffee break finally. And there is another important point for experimentalists. If you want to, I mean, the methodology, maybe maybe there's a bug somewhere, or we have to check that, but apparently it's working pretty well. But there's a practical point. How do you choose lambda? Because as you have seen before, if you don't choose the right one, it's going really down. You lose 70% of the performance if you just miss the right lambda. And there's no, I mean, in the theory, there's nothing that suggests how you should choose the lambda. And one could maybe rely to some theoretical distribution of the link distribution. So here you see that when lambda is very small, you have fewer short links and more longer links. Then you get finally the good result. And if lambda is very high, you lose many connections, and so you have this distribution which is really jumping up and down. But still, I mean, you require too much, probably, the theoretical distribution of a network which is maybe not following the theoretical distribution. There is actually another principle that one can exploit, what I call here the Dalton principle. At the mature stage, neurons are either excitatory or inhibitory, so real excitatory and real inhibitory neurons, meaning that the excitatory neuron sends out only excitatory connections, and inhibitory one only inhibitory ones. So what happens if lambda is very small? You have a lot of connections, and you have a lot of violations of the Dalton principle. The more you increase the lambda, the more you approach the perfect reconstruction of the network. This is what happens here. So with small lambda, you go up, you increase lambda, and then finally you get to the plateau where you have only excitatory or only inhibitory connections at the right place. Then what you can do is to compute the lambda relative to the plateau, so when you finally get the value 1. And you can do that for multiple simulations. So here I did it for many different networks. And what you find is that if you compare it to the optimum, so the peak, which was obtained, of course, comparing it to the structural connectome, what you find is if you define the lambda, the right lambda as the one of the plateau, it's always lowered compared to the peak MCC. And that basically tells you that the methodology is getting right, first of all, the sign of the connections, and then it's refining the connectome by pruning the connections that are not needed, right? And, well, of course, then one could elaborate a little bit on this. Maybe instead of taking the first point of the plateau, the second one, but, I mean, this is something we can still work on. Just for the sake of knowing what's happening now is with just choosing the first point of the plateau, what you get is the excitatory connections have been retrieved quite perfectly, so 0.98. The inhibitory one is a little bit less, and I have no explanation for that for the moment. We have still our results from the last few days, so I'm still trying to understand what happens. And this is basically closing my talk. This is a joint work with a master student who graduated a few months ago, and 2 mathematicians from the University of Milan. Of course, I would be very happy to play and to have this type of data in order to understand if the methodology can be really brought to the I mean, to the applications. And well, this is just to apologize for the fact that I had a talk which is completely different with respect to the session, but we have here a poster by Gianluca, which is more related to TVB and the macro scale, and he's dealing with investigating the impact of cortical lesions on brain complexity. So it's still PCI stories in a TVB model of stroke. And I finish.